Hello friends, let me adjust the microphone. This will be a slightly different video from what you're used to seeing on my channel. This will be a video about the phylogeny of DRD2 gene. I picked out some variations from DRD2 gene and I decided to research the relationship between them. So the software and the samples used for this video is my phylogeny finder. Which is a tool I sort of haven't really finished developing yet. It's working, I got it working, but it's not entirely doing everything I needed to do. So it's gonna be a little bit before this tool actually is able to be released. Before I can release this tool, it's gonna be a little bit. But here it is. I might never release it publicly, actually. I don't know, because it's just so much work and it's... maybe I should sell it instead of releasing it publicly. It's developed in C++. That's right, I'm using a really old language to develop it. Originally, I tried to do it in Python, but I realized I don't have the skill in Python to do it, so I just went back to the C++ and did it there. Because I do actually have pretty decent ability in C++. The samples I used for this project are from OpenSNP. It's 592 people. 191. I can show you the exact individuals here. These are the individuals I used for this project. It's 591 people. A lot of people. A lot of people, right? And here are the SNPs that this video is about. Basically, the relationship between these four SNPs, how they're typically inherited, what's typically inherited together with what. That's what it's all about. We're making a phylogenetic tree. Now, with a gene like DRD2, these SNPs are all fairly far apart from each other. And these mutations happened a long time ago, which means there's a lot of dislinkages, which means that when you make a tool like what I've done, it's going to be a statistical reality rather than an objective reality. It's going to be, well, in 70% of the cases, there is this phylogeny that we can observe. And in another 15% cases, this phylogeny is broken, stuff like that, right? It's not 100% precise. It's not 100% accurate because there's dislinkages, and because all of these SNPs, all of these mutations are really old. If I were to do a similar project with HERC2 and OCA2, you would see that the statistics are a lot different. But let's go ahead and look at the actual results, which are here. So as you can see, what this tool did is it went through, it looked for these SNPs, these four SNPs in DRD2, which are Pro-Frenetin Pro, this we all know. There is this variation, which I called grass. It doesn't have a name. It's just RS46, 48, 317, but I'm going to call it grass because it's a lot easier to refer to it that way. There is TAC1, which we all know. And then there is RS10, 76, 560, which is really difficult to pronounce. So we're going to call it GUI. The A allele in the GI is typically associated with an increased risk of alcoholism, and the A allele in grass, let's remember it, is typically associated with an increase in the risk of schizophrenia. Now, what's interesting is just observing this, you will see that in every variation aside from grass, the derived allele typically decreases the availability of dopamine D2 receptor sites. The only exception to this is grass. So we can see here all of the data that was collected by my phylogeny maker. A lot of data. 590 something lines of data. So it looks like here for GUI it was 590 genotypes. For Pro-Frenetin Pro it was 590. For grass it was 590, and for TAC1 it was unfortunately 586. So it seems that some files have grass and GUI and Pro-Frenetin Pro, but not TAC1, which is unfortunate, very unfortunate. Now, as you can see, the derived allele frequency stuff, this statistic is, I wasn't able to do it. Something isn't working, I don't know, I'll figure it out, I'll fix it. But what is really interesting here is the A-scores. The A-scores measure how closely related the two genotypes are, which means how statistically significant is the relationship between a particular genotype in one SNP together with a particular genotype in another SNP. And the higher the A score, the better. So it can be negative, which means there's a negative correlation, or it can be positive, which means there's a positive correlation. And the higher the number is, generally the better the correlation is. That's what it is. The higher the number, the better the correlation. Right, so it seems that the highest correlation is between TAC1A and this is, I think, GUI-A. So the highest correlation is between these two genotypes, having these two alleles, the A allele in TAC1 and the A allele in GUI. And what's interesting about these two is that both of them actually decrease dopamine D2 receptor sites availability. TAC1 and GUI both decrease dopamine D2 receptor sites availability. Actually, the derived allele in both is implicated in alcoholism too. So it's kind of interesting that not only are they similar in what they do, they are also similar in how they are inherited. They are probably very close to each other, actually, if you think about it. Yeah, they probably are. Although TAC1 is in A and CAR-D gene, technically. It's not even technically DRD2. It's really close to DRD2, but it's technically outside of the DRD2 scope. The second closest relation was between, this looks like GRAS A, and this looks like, what is this? I forgot, GUI. Yeah, GUI-A. So this is kind of interesting because the A allele in GRAS increases dopamine D2 receptor sites availability, and it's typically inherited, or at least statistically speaking, it's likely to be inherited, together with the A allele in GUI, which decreases dopamine D2 receptor sites availability. That's very interesting. That's very interesting. And finally, the third strongest correlation is between, this looks like GRAS A and TAC1A, right? So the A allele in GRAS also is kind of strongly linked to the A allele in TAC1, which is once again kind of interesting and contradictory because the A allele in GRAS increases dopamine D2 receptor sites availability, whereas the A allele in TAC1 does the opposite. It decreases the dopamine D2 receptor sites availability. Now, when it comes to the derived allele in prophenatine pro, it seems to have a negative correlation with every other derived allele. For example, the derived allele in prophenatine pro seems to have a negative correlation with A in GRAS. So having derived allele in prophenatine pro will technically, usually it will go together with the G allele in GRAS, or at least statistically speaking, it's more likely to go together with the G allele in GRAS. Now, the A allele in prophenatine pro is also statistically not likely to go together with the A allele in TAC1. Once again, it's technically usually going with the G allele in TAC1, which is the ancestral allele. And the derived or the A allele in prophenatine pro typically goes with it looks like the C allele in GUI. However, there the interactions, the A scores for the interactions are not particularly impressive. They're negative, but they aren't all that negative. So it looks like prophenatine pro actually doesn't have that close of a link with all the other SNPs that are used here in this comparison. It seems that the closest link is between TAC1 and this is GUI, right? TAC1 and GUI, GRAS and GUI, and GRAS and TAC1. It looks like TAC1, GRAS, and GUI technically, usually, typically, I say technically a lot, but I really mean typically. It seems that these two variants, these three variants, are typically inherited together. Well, thanks for watching my little video. Leave a like and subscribe. I will make this better. You will see a lot more content with this phylogeny maker.